Hello everyone, welcome to the course Python for Biological Data Analysis. Today we are going to discuss Chapter 6 Pandas for Data Analysis. I am Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Let's start. Okay, so first of all, you have to open the Chrome browser because we are working on in the Google Colab. So write Google Colab, click here. Now click GitHub and type here in the search box H100. Press the enter button. You will see this kind of window. Click here, scroll down and select Python for Life Sciences. Now scroll down, you will see Chapter 6, Pandas for Data Analysis. Click here. This interface will be open for you, and now you can connect this notebook with your internet. Press connect. Okay, so today we are going to discuss Pandas for data analysis. This is my brief introduction and the introduction of this course. These are the course outlines. I have covered the chapter first, second, third, fourth, and five. Today we are in chapter six. If you want to watch these other lectures, just click here and it will lead you to the corresponding lecture. Okay. In Pandas for data analysis, in this chapter, we will talk about the data frames for structured biological data, data cleaning and data manipulation, some filtration and grouping experiments, and some handling of missing data. Okay, now welcome to this chapter using pandas. What actually panda is? In last video or in last lecture, we have discussed the NumPy or the numerical Python library, and today we are going to discuss Pandas. It is another powerful Python library and it works with structured data. In biology, we often deal with the structured data in the form of tabular data. From the tabular data, I mean the data that contain the numbers and values and in the form of Excel sheet. And these data could be of gene expression, matrices, protein interactions, and clinical trials, and etc. etc. So we will learn today some basics that how Panda we can use Panda and how it is helping us. Okay. So first of all, I have included this installation setup. Although if you do not use this installation setup, it will also work. But I have included. Okay. So before we begin, in our last video, we import a NumPy, but today we will import NumPy, Seaborn for visualization, and Pandas for data manipulation. So if you read this script, this is the install command, and this is the I'm using pipe for the installation. So pipe install Panda Seaborn. In case you are using this tutorial in your personal computer, not on the Google Colab, then you don't need to put this exclamation mark. You have to remove it. This exclamation mark is particular with the Google Colab usage. Okay, so let's run this cell. It will install the pandas, it will install the Seaborn, and it will import these three uh, libraries. Okay, Pandas, Seaborn, and NumPy. And it says requirements already satisfied, so that's okay. Now you can clear this output if you don't like such kind of output. Okay, what actually is a data frame? A data frame is just like an Excel spreadsheet, but in Python. The Excel sheet we are looking in our computer, that is an Excel sheet, but if you look that Excel sheet or some data pieces in Python, so we can call it a data frame, or we also in Python they also call it DF, just an kind of abbreviation. So each row in our data normally represents kind of some observation for a gene, for protein concentration, for some for anything else. But if you are aware of the Excel sheet, you must be, I assume, then there are different rows and different columns. So each row records some observations, and each column is a kind of variable like the column of age, the column of sex, the column of, let's say, living area, the column of let's say, salary. So these all are different variables for a particular person or for a particular object. Okay, so uh, let's simulate a gene expression data set. For example, we have a gene, it has some name, and the data set contain expression level, and it contain the tissue information, and it contain the conditions like the patient or the data is from the healthy sample or from the cancerous or cancerous patient or cancerous sample. Okay, so I'm generating a sample data set first. So data, and then the is equal to, and then I used curly brackets. Now I will insert gene, expression level, tissue, and condition. For gene, I included the TP53, BRCA1, EGFR, MYC, and CDK2. For the expression level, I just randomly dropped some values, and for the tissue, liver, breast, lung, brain, and again liver, and healthy, cancerous, cancer, healthy, cancer, just a kind of data. Let me remind you that you don't need to make your data every time. Most of the time, we use a data from the Excel and we just uploaded that data directly. But here, I tried to create some data. First, a simple kind of data, and then will teach you how we can manipulate. But if you upload your data set, so that is also okay. The program will read it, and you can work. You can use pandas for different kind of manipulation and things. Okay, so and then the df is equal to td dot data frame data. Okay, this is actually this variable, this is a data, so that shows our data, so this is our data. And if we upload our data set, then it will be like this, data is equal to read.csv, that kind of command. So right now we have made our data set just like this. Now, yesterday or in my previous lecture, we discussed about the NP, like the NumPy function. So today we are going to discuss the pd.dataFrame. So this is actually a Panda, the function from the Panda library. And this data is actually this variable. So we save it, this data as a DF, as a data frame. And now we want to print the data frame. So we will just write DF. So once we click here, it will run and it will provide our data in this form: gene column, expression level column, tissue column, and condition column. Okay, now once you have some data set or you generate it or you upload it, then you need for some sort of handling or some sort of cleaning. So in handling, for example, we often need to rename columns for clarity. For example, you got some data from me, from some organization, and their column names are different. So you will need to change those columns as per your need, for your understanding. So this is called data handling. So let's learn how can we rename columns for clarity, how can we normalize values. So in this lecture series, I will use the normalization process by dividing each value with the maximum expression, because I'm using the expression level. So and then I can also standardize the labels. For example, if I want to change the liver label to the hepatic, so that is a standard term, so that is why we call it standardization of the labels. Okay, so now I have included these three steps in one. So how we will rename the column for clarity? df.rename, this is another function. Here we use df.dataframe. Now we are using df.rename, then smaller bracket or parenthesis, and then I want to rename one something from the column. These are four columns. So I want to rename something from the column. Then we will put is equal to sign and the curly bracket. What I want to rename? I want to rename expression level to the expr. You can see we have expression level. Now let's say I want it expr, and in place is equal to true. That means change it to the expr and show me, change this label to the expr. Okay, for add a normalization expression to the column. In my previous lecture, I mentioned to you that in NumPy, also in DF, we get escape from the definition and while loop and for loop, like in my previous four lectures. I showed you that for too complex. So that's why the Panda and NumPy are pretty much easy and straightforward compared to the normal Python. So df now ex, it means that add another column for the normalized expression and name it expr underscore norm, for like expression normalized, and that will be equal to the df, which df, the expr df, and divided by the expr, but the maximum value of expr, as I told you that the normalization will be provided by the division of the maximum value. Now also let's create another data set or change another thing. The tissue, so the df tissue replace in the tissue column, this one replace liver to hepatic, and then print that uh, data frame. So click here. Now you can see gene expr, the expression level is now changed to expr. In tissue, you cannot see liver, now it is hepatic. In conditions, they are similar. We did not touch anything, and another column has been generated, that is the normalized expression. So quickly we were able to do or to create this another column with the data. So now you can think, for example, you have a data set that is composed of, let's say, 1 million entries, and you want to calculate the expression normalization column. So you can easily do it or quickly do it in with the pandas. Okay. Now let's come to the filtering and grouping section. For example, or we often want to focus on some specific condition. For example, I am focused to cancer samples in a big data that contain, let's say, 1 million lines or 1 million rows. Then I want to extract or group the cancer samples differently. So, or then let's say I'm also interested to compare different tissue with each other. So these are two different tasks. So I call it both these tasks will be from the grouping section. And this is also called filtration. For example, we are filtering the whole data based on the cancer or based on the tissue. So let's see how we will do. We will generate another DF data frame. Its name will be cancer DF, and DF dot query is equal to condition. I put the condition that will be cancer. So that means that generate another DF cancer underscore DF and activate this condition cancer. Now I'm also interested to group the group use the function group by tissue and mention or compare the expression level with mean and max value. Okay, let's run it. Now you can see brain, breast, hepatic, and lung. Okay, there is a slight mistake because I used two different uh, data frame, like group data frame and then cancer DF here. But I am seeing the latest one, the grouped one. So let's separate this thing and you will see this in a separate form. Let's say click here. Okay, you will not see this. It will be like this now. Okay, filter for cancer sample. Okay, play. Now you can see that from the data set we extracted only based on the condition column because this was the condition column. So we extract only the cancer samples or those samples which satisfy this condition or this column. Now run this thing. You can see based on the tissue we just extract or kind of comparison. So for the brain, the mean value of expression is 6.3 and the max is also 6.3. For the breast, 5.5. For the hepatic, 6.05 and the max is 7.2. For the lung, 8.10 and 8.1. So that means very quickly we can manage or we can drag some meaningful insights from the big data or big data sets. Okay. Uh, Now, how we will handle the missing data in biology? So, missing data, when we use a data set collected from the lab experiments or from somewhere else, so normally we remove the missing value. So here I'm going to generate a data set having some missing values. Let's say my data set is the name of my data set will be missing data. And like in the first step, we generated for different genes and expression level. So here we have four columns: sample ID, P1 to P4, age, 35, 42, none. So none is actually the missing value, and 58 biomarker A. Also, I placed one none value or missing value here, and biomarker B is complete. If you go with this kind of data set in your analysis, you will always have some error of none error. We call it none error. So that's why the good choice is to clean your data from the missing values. Now, uh, let's say, uh, okay, now if you see the original data frame with missing data, so if you see there is one missing value in the none, in the age, and one missing value in the biomarker we already created here, biomarker A. So please try to understand here this concept. There are two options you can treat the missing value in your data set. First one is to drop them out. For example, you have 1 million rows in your data set, and it's a big data set, and there are few missing values. You print it here, and you cite like these are very few, and you can afford dropping them out from the data set, so that's okay, drop them out. But if you don't want to drop them out, or you cannot afford them to drop them out, then in that case, the researcher normally keep that value with the mean value of that column. For example, I will give you a normal example like we have 1000 rows. Let's suppose in this case we have 4, 1, 2, 3, 4. Let's say we have 1000 rows in a data set, but one value is a missing value here. Now, I don't want to drop this data or this row from the data set. In that case, normally we keep mean of this whole column age, take the mean of this age column and keep that mean value here. So this is another option, and you have to think twice or thrice before exercising dropping or filling the missing data. So in this case, I'm going to drop these missing rows. So we will have two rows left, one this one complete and this one is complete. These two will be missing out. So I am generating df.drop and missing underscore df dot drop na is for the missing value. So I'm using this function, and let's run it so you can see those two values or those two rows were dropped out. In this case, I'm using filling the missing data. For example, I'm using the mean or age. So missing df is age and biomarker A. And here I asked like for the age, put the mean value, and for the biomarker, put the median value. So you can also keep this median mean, or you can also keep this mean median. That is up to you to decide. And you can see now we are safe, all our data is safe, we did not exclude anything, and we just added some uh, we just filled that missing gaps. Okay, that was for the data, for the pandas, for the data analysis. So if you are dealing a bigger or larger data set, so you will need these essential steps to go on further for analysis. So I hope you got some idea from this notebook. And if you are liking my content, please thumbs up and share with your other colleagues and students. And stay tuned. Normally, I'm busy with this course, so I'm not bringing the other stuff like docking, simulation, protein design, and gene expression. Too much things are remaining or left for me. I have a long list, so subscribe to this channel. You will have much more interesting things ahead. Okay, thank you so much. Bye bye.